Hello. So it's my pleasure to introduce our feature speak for the night. Tamara Munzner is professor at UBC Department of Computer Science since 2002. And she's been active in visualization research since '91. So long history of research and a lot of experience. And she's been and she got her PhD from Stanford. So her wide range of applications, I'm just going to list it here, including genomics, evolution biology, geometric topology, computational linguistics, large-scale system administration, weblog analysis, and journalism. She's co-chaired several notable large visualization meetings, InfoVis in 2003, 2004, and Euroviz in 2009 and 2010. She's also the founding sort of executive committee member for the BioVis organization. She recently, last year, published a book called Visualization Analysis and Design, and today she's going to tell us a bit about her book, and with a specific focus, of course, in biology. So title today is Visualization Analysis and Design in Biology. So welcome, Tamara.
Thanks. So visualization analysis and design for biology. Now, let me check. I tend to move around a lot. Am I moving in and out of the sphere of the mic? So, attempting to stay rock solid. I will try to, if I'm going in and out of mic, wave your hands in the back, and I will try to be less distracting in that way. So I'll start out with giving you some context for visualization, how I think about it, and then move on to actually walk through 3 different examples of projects we've done in my lab in the past little while, and then do a bit of wrap-up. So let me start with a definition of visualization and why you might care about it. So I think there's a central question with visualization is why is it useful to have a human in the loop to do data analysis? And my definition of visualization, I'm going to abbreviate that as VIS for most of this talk, saving many syllables over the next hour. So computer-based VIS system provides visual representations of data sets designed to help people carry out tasks more effectively. Now, why do I feel the need to have data and people in the same sentence? What is the point here? I would argue that you need VIS when you want to augment people rather than simply replacing them with some computational decision. So it's a natural question to say, when would this be? And what's interesting is, for those of you who have a computer science background, of course, you could write a program to solve many, many problems. So the issue is, what if you don't know exactly which problem you're solving in advance? There are many possible problems. And so, let me get this down a bit. If you already have a fully automatic solution and the people who need to run that trust the solution, then you don't need VIS. But there's a lot of situations where that's not true. So I'm curious, how many people in the audience have seen Anscombe's Quartet? Everyone who's been in my Viz class, yes, and a few others as well. So I love this example. It was designed by the statistician Anscombe in the early 1970s. And it's a set of 4 data sets. They're small, and they have identical summary statistics with some basics of mean and variance in X and Y, and an X and Y correlation. Now, the instant I show you a picture of these, we see that the world is not the same in these 4 data sets. One looks like what you might think given these summary stats. One is clearly nonlinear versus linear. And of course, there's methods for dealing with nonlinear data, but you have to know to use them. There's one where that regression line is a bit off because of an outlier. There's one where the regression line is completely terrible and pessimal because, in fact, this was carefully designed to mislead. Now, you might say, well, okay, this was a toy data set. But life only gets worse. As you get more points, as they are dynamically changing over time, as you have heterogeneous data where you don't just have a single table but a lot of things putting together, there gets to be a time when having a human in the loop to look at what's happening is very useful for doing visual data analysis. So here's another question. Why are we even doing this thing of having a visual representation? What good is it doing? And visual representations are a special case of the more general example of external representation. And when I say external, I mean outside of a human's head. So what is the point of trying to get out of a human's head? And the idea is that although in some ways humans are amazing, we're great at pattern detection, we have very good long-term memory, There's a way in which we have pretty finite resources in terms of short and medium-term visual working memory. And so any time that we can replace something that's cognitive with something that's perceptual, we free up these resources to do something interesting, like, say, analyze biological data, rather than do something really, really low level. So let's look at an example of what I mean by replacing cognition with perception. Here's a table of numbers. They might be dear to your hearts. These are actually gene expression levels measured with the number of genes at different time points in different experimental conditions. Now, if this font were big enough that you could see it in the back of the room, and I asked you a question like, tell me the distribution of numbers in these tables, roughly positives versus negatives. Now, it's not that you can't read numbers. You can all do that. I have faith. But you read a number, you store it in your head. You read the next number, you store that in their head. You start to compare. You read the next number, you store that in their head. Soon we start to run out of storage locations for this kind of short-term memory. You're doing something cognitive by doing that reading and comparing. What I want to do is instead exploit the human perceptual system. So in this case, what I've done is I've encoded those numbers as a diverging red-green color map, and I've color-coded them. And then the ability to look at these distributions quickly is something that you can do. And you can notice that along the top, you have a fair amount of red, a mix of red and green, and then a mix. Whereas along the bottom, in that third one, we've actually got quite a bit more red in that LPS4 case. It's an example of where you've got a different distribution. In this case, you can conjecture that the experiment was actually successful in this case, reducing the gene expression levels. So this is an example of where we have replaced something cognitive with something perceptual. That's going to be the underlying theme, except often in a little more detail in the rest of this talk. So what I'm going to do is try to get you thinking about visualization in terms of a very analysis-oriented way of thinking. How can we take a visualization and chop it apart into pieces and think about those pieces in a useful way? And so what I found useful to think about is this framework where there's 4 levels and 3 questions. So let's walk through those. What I'm going to call the domain situation, in my language, you guys are a domain. So biologists are one domain. Financial analysts would be another domain. People trying to look at their music listening habits over the last year on Last.fm would be another domain. So people who have data and some questions to ask about that data. So from a viz designer's point of view, and I'm going to speak to you as a viz designer, I'm going to say, so who are my target users? What are their needs? What are they trying to do? Why are they bothering to look at this data? Now, what's useful is to say that there are things that cross domains. There might be something extremely similar if a biologist is doing a particular kind of data analyst and a financial analyst for a company. They might be looking at data that at some level is similar, even though they use radically different words to talk about it. And their tasks might be similar, even though the words that come out of their mouths don't actually directly seem to map up. But if you abstract them from the specifics of the vocabulary language to a unified way of thinking about it that we can address using visualization, then suddenly we can actually start to think in a way that doesn't mean we're doing one-offs for every single problem in the universe. We're thinking in a more systematic way. So this step of abstraction is really, really important and crucial. And I'm going to focus on 2 big ones. What is being shown? I'll call that the data abstraction. And what's interesting and hard about this is you might think, well, how hard could it be to abstract the data? It turns out there are some challenges in translating. But there's not just translation, there's also transformation. You might take the data that you're given and do some kind of transformation to some other form such that when you draw that, it actually helps them solve their problem. So the simple case is when they hand you the data and you simply draw that data. You often do a series of transformations along the way so that you can actually end up drawing them a picture that's useful and helpful. And those transformations might be in the course of an interactive analysis loop. The other question we're going to ask is, why is the user looking at that data? What is their task? I'll call that the task abstraction. And again, that's something where we have to go from domain language into something that we can think of in a unified way to address with visualization. So the what and the why are at this level of abstraction. Well, then we have to design what I'll call an idiom. You might see this called a technique or a method or approach or some word of that sort, where I want to emphasize, again, 2 separable questions. One is, how is it shown? And what I'll really think about is, how do you draw that picture? I'll call that the visual encoding idiom. And then how do you manipulate that picture? Because we often don't just have a static picture. We've got some kind of dynamic thing that we're interacting with it. Now, I've separated out the idea of idiom from the algorithm level, which is specifically given this idiom that you've designed for how to draw and how to interact. Now, how do we program that efficiently on a computer? So the algorithm level I've separated out from this. Now, you might wonder why I have proposed these 4 levels. And they're actually motivated by this extremely tricky problem of figuring out, have you achieved victory? Did your viz solve the problem that you intended it to? And it turns out that the methods you need to use to analyze this depend a lot on these 4 different levels of design. So while I'm not going to walk through this in huge detail, let me at least give you some glimmerings of what I'm getting at here. So let me start with the algorithm level. So the methods of computer science are fantastic for analyzing algorithms. We, as computer scientists, are typically trained, how can you do computational benchmarks that are useful and meaningful? We're going to measure the system time, the system memory, and if we're slightly more theoretically inclined, we might also analyze the computational complexity. So figuring out if your algorithm is doing the right thing is something that computer scientists are typically well-trained in doing. What about this question of designing stuff that's supposed to exploit the human perceptual system? Well, I use the word design. We're often going to take methods out of the design field, in fact, of trying to understand, have we actually considered the space of alternatives enough? And in order to figure out if that design actually works with the human perceptual system, we're often going to run controlled experiments in a lab using methods straight out of cognitive psychology, where we're measuring human ability to actually conduct some task that you tell them to do, and we're going to measure them with a stopwatch and see how many errors they make, and try to figure out, is this working given human perception? Now, what's interesting is people often get a little confused about these 2 levels. And so if you propose a new way of encoding information designed for human, and then you tell me how fast your algorithm runs, you've convinced me at the algorithm level, but you have not convinced me at the idiom level. So you have to be very careful about validating appropriately for the claim, because telling me how scalable or fast your algorithm is doesn't help convince me that it's helping humans. Now there's even one more thing that gets tricky here. Did we understand that task correctly? Did we actually watch what people are doing in their daily lives? We actually think about things like field work, where we're taking methods out of anthropology and ethnography. So when we as viz designers talk about field work, we typically don't mean the jungles of Borneo. We mean we go into your labs and watch you. And we see, first, we do talk to people, and we also observe them and try to get a sense of what are they doing in their labs. And then try to figure out, if we actually deploy the tool in a realistic setting, are you using it in the way that we had in mind? And it turns out, here's another thing that a lot of viz papers sometimes get wrong. You can't prove that you got the abstraction level right through a controlled experiment in a lab, because you're telling people what to do in the experiment when you measure and time their responses. We want to see what happens in the wild real-world setting, where we actually deploy, and we want to see how it's used in terms of real data analysis. So it's interesting to note that these are a pretty broad set of methods across different domains, which many people are not necessarily trained in all of these. So there's some interdisciplinary things going on in viz of trying to address these different levels with appropriate methods that actually convince us that you have solved the problem you're trying to solve. Let me mention briefly, usually no single viz project is addressing deeply all of these layers in terms of innovation and research contribution. So often in a given research paper, you don't validate at all of these levels at once. You pick the one that's suitable for the research contribution you're claiming. Otherwise, our papers would take 17 years to write, and they would be 200 pages long, and both of those would be unfortunate. So be aware that you won't see all of these in any paper, but hopefully, if it's a strong paper, you'll see at least one of these validation methods, and it will be matched to the research contribution. So let me mention there's some major streams of work in visualization, and in some cases people say all of those higher levels above the algorithm level, those have been established by other people in other papers. And I'm going to just allude to those by referencing them, and what I'm going to focus on here is, let me design a better algorithm. Let me scale up to larger data sets, let me handle more heterogeneous data sets, let me do something faster. So there's a lot of work, and I'll call that technique-driven work, which is exactly aimed at the algorithm level. And of course, in bioinformatics, there's an enormous amount of work at the algorithm level. The other way of working, which is I think a lot more common in Viz than in most bioinformatics, is starting from this question of here is a deriving problem, there's a real-world user, here's their data, here's their task, let me figure out what they need and go through this process of actually trying to abstract and come up with appropriate idioms. And sometimes then there will be major algorithmic work to do, or sometimes there won't be too much deep you have to do with algorithms, and a lot of the intellectual value is trying to do that task and requirements analysis and understand that perceptual design space. So in my lab, I actually strongly feel that it's great to try to get all of these angles of attack. You can do technique-driven work and problem-driven work and learn. You can have an idea on evaluating and trying to characterize when these methods are good, even not just on the systems you've built, but more abstractly trying to characterize this design space, and then try to feed back into the theoretical foundations, because at some level, the reason I, as a Viz person, am designing tools for you, not only because I love you, but because watching you use these tools will help me understand more about how to do visualization. And so it's feeding back into my own research field, not just building tools as a service. So let me mention briefly, to give you a sense of the kinds of things we've done in my lab, I have a special spot in my heart for networks. That's the kind of data I used quite a lot in my PhD. So we've done a lot on trying to scale up, both for general node-linked networks and trees in particular. I'll talk a bit later on about the tree juxtaposer system. I've also done a lot of work in dimensionality reduction, going from very high-dimensional data sets down to some lower-dimensional structure that captures a lot of the variance. So we've done a mix of algorithmic work and systems building work in that. I'm not going to talk about that today, but if folks are curious, then grab me afterwards. We've also had this question of, so let's try to evaluate, in the case of these methods, how should you show that data after you've calculated the dimensionally reduced data? So a mix of actual perceptual studies in the lab and expert evaluation of when are we actually succeeding in showing people the structure of this data. We've also done a fair amount of experimental work of these human-centric experiments in the lab to try to compare different techniques for visualizing data. Again, not going to talk about those in much detail today at all. I've done quite a bit of work on trying to take this idea of there's principles of design and how can we think about them, and there's been quite a bit of that kind of theoretical foundations work in my group. We'll mention culminating in a book on visualization analysis and design where I really tried to bring a lot of things together. And this talk is going to be structured around that framework out of that book. And as he also mentioned in the intro, I've done this sort of problem-driven work for a lot of different flavors of people. Folks at AT&T doing large-scale system management, fisheries management where they're simulating how much fish are caught versus allowed to go out and breed over 100 years, and then helping people set fisheries policies. Overlay networks for the people actually designing cars at BMW about how do you actually get the signals to go from when you hit the little button to make the window go down to actually having that motor go in ways that, of course, don't overload things like hitting the brake pedal and having the brakes go. Investigative journalism. How do we design systems for letting people look at data sets of tens of thousands of documents and not have to read every single one to make sense of them? But what I'm going to focus on today is the work specifically in genomics, and I'm going to pick two of these systems to talk about in more detail, and I'll just mention that there's several more if folks are curious, and a lot of them do have videos and papers available on the website, and I'll have the URL for that at the end of the talk. So before I go into that kind of problem-driven work, let me just mention a methodological question of how do you even do this work? And I've ended up reflecting on this enough that we've written a paper to try to provide some prescriptive guidance to people about how do you do this sort of problem-driven work. It turns out it is tricky, and there are many ways to go wrong. And so between the three of us, two of my former postdocs and me, we've done about 21 of these systems, and we tried to figure out what are some ways that would help people do them faster and better in the future, and what are some things we did wrong, or papers that we have reviewed have done wrong, or papers that we've seen in the literature to try to keep people from falling into these pitfalls. And again, to notice about this idea of abstractions versus domains, these are from a lot of different areas. Yet there's some that have pretty similar visual appearances and some that are quite different. And so it's not that all the genomics ones look identical and then the car ones look different. It actually comes down to questions like, well, what are the data and task? So there's these interesting cross-cutting similarities and differences when we think about these at the abstract level, not just the domain level. But I just want to mention one idea out of this paper, and it alludes back to the question of when would you even do visualization? Because I think that's an important question for audiences like this that are not primarily Viz people. They're people doing something else that are thinking, when should I think about Viz? When might this be useful for my lab? And so we found this, it's a very theoretical abstract diagram, but it's useful to think, when should you use design studies to do problem-driven Viz research or even visualization at all? And I find it useful to think about how much do you understand about the task and where is crucial information? And what I like to think about is along the bottom axis there is crucial information in the heads of users only, in which case there is no hope of doing computation, or is more and more of that actually in computable form? But not just the information, but the task. How, as the designer of a system, do you understand their task well, or is it actually quite fuzzy? And it turns out that if you exactly understand their task well and all the data is computable, that's a great sweet spot for automating things, whether it's with a custom program or using some kind of machine learning approach to learn some parameters. But there's this big gray area in the middle where one of your goals is to understand more about that analysis task and to actually come up with a much more crisp definition of that. And that's often where visualization is really well suited. So I want to give a plug. Even if you never do visualization for the rest of your life, it would be great if folks in bioinformatics thought about this paradigm of problem-driven work, of engaging heavily with the target end users, of doing some of these flavors of human-centered design that a lot of folks in human-computer interaction have thought about. How is it that you could design tools well-suited for users? So I think that bioinformatics is really ripe for exploiting this kind of methodology far beyond visualization. So this question of understanding what a group of people need, there's all kinds of fabulous work which shows that asking them is necessary but not sufficient. That a lot of retrospective introspection about how they spent their time is only a partial story of what they actually do compared to observing and trying to do iterative prototyping to build tools and then actually see how they're being used. So as you're hearing me talk about the Viz, even if you don't yourself intend to ever do Viz, think about how this kind of methods might help you with your own non-Viz tasks as well. And of course, if you care about Viz, then this is the talk for you as well. So with all that as introduction, now let's actually talk about some of these projects. So the Cerebral System was actually joint work with the Robert Hancock lab. Back when Jen Gardy was there, she's now actually got her own lab as well. And this was joint with Aaron Barsky, a student of mine, and also Robert Kincaid from Agilent. So this is one of these interactive Viz systems which is much easier to understand after you've actually seen a video in action. So before I tell you much more about it, I'm actually going to show you a video.
Cerebral is a tool for systems biologists to visualize and explore quantitative data from multiple experimental conditions. The data is overlaid on a biomolecular interaction graph, laid out using annotations about the location of the biomolecule in the cell. Cerebral is implemented as a plugin for Cytoscape, a tool which loads biomolecular interaction graph models and associated node and edge data from multiple sources. Our plugin replaces the standard graph viewer. Many layouts optimize graph topology but ignore biological metadata. Here, the colors show the membrane-bound compartment where a biomolecule is located in a living cell. Molecules from different compartments are mixed together in the graph view. In contrast, Cerebral divides the nodes into layers according to compartment in a familiar cross-sectional view of the cell common in cellular biology diagrams. For immunology data, proteins activated in response to infection are shown in a special layer below the nucleus, clustered together into groups with shared biological function. Biologists can position nodes by hand and pin them to form a skeleton of important biomolecules. The biologist views experimental data by selecting node attributes corresponding to measurements collected for each node in the graph. Each experimental condition is mapped to a small multiple view and an axis in the parallel coordinates view. The values associated with a node map to an intersection point on the axis and a color coding in the small multiples view. The size and number of columns in the small multiples view is adjustable. The user can reorder conditions by dragging axes or the small multiple views. The reordering is linked between views. We reorder the values to place the time series in order and each control LPS condition beside the treatment LL37 condition. Looking at the small multiples, the LL37 treatment has the largest effect at hour 4. We compute a direct calculated difference in the main view and see that most genes are colored orange, indicating repression, particularly amongst the inflammation-causing cytokines and chemokines. We can examine the same data set in the much larger map kinase interaction graph. Here we use mouse hover highlighting and linked panning and zooming to explore a more complex interaction model.
So I'm going to stop that there. That's enough to give you a flavor of what the system feels like to use.
Looking at the small multiples, the LL37...
Sorry, I didn't realize I'd embedded that in the slides. So now that you have a sense of the look and feel, let me talk about some of these design decisions. So first of all, what is the data abstraction? What is the language of Viz that I'm going to use to talk about this data with? So in terms of the data set type, well, I've got network data. So in this case, I've got nodes, which are genes, and we've got these known interactions between them as links. So I'm showing that explicitly as a network. And I've also got tabular data. So I've got a table with these quantitative attributes of, for every gene and experimental condition, what is the gene expression level that was measured, and also a few non-quantitative categorical attributes like where within the cell did that interaction occur, what are known functional groups. So I've got this mix of attributes, and it's usually really important in Viz to understand whether your data has an intrinsic ordering that should be matched with a way of showing it that's intrinsically perceptually ordered, or whether it's simply a nominal categorical data saying it is this but it's not that, in which case you don't want to misleadingly communicate order information with the visual channel that you're using. So let's talk about the task. Now, the task is to try to interpret the experimental results of microdata with respect to what's already known about some gene interaction network. And so people often think about visualization as being designed to help people have their eureka moment of doing something they could not have done before without the tool. And that is fantastic when it happens, but it's not actually our bread and butter. A lot of what happens in Viz is we're trying to take an existing workflow and make it faster. So to try to have something take 10 minutes instead of 4 hours is a typical kind of success case. So we're trying to take what they're doing and help them do it faster. So what are we doing abstractly? Well, they were comparing distributions between these attributes and trying to interpret those in the context of this known topological structure. So if you think about actions, you're comparing across some attributes and trying to understand that network topological structure, and it's for the goal of discovering something you didn't already know, as opposed to, say, presenting already known results. So let's talk about how. So for reasons that if we had much longer I could go into, but I'm not going to go deep into today, arranging space to communicate information is the strongest of the human perceptual cues. So this question of how do you arrange space is really central when we analyze a visualization. So we've introduced this custom way of laying out a node link diagram. So we saw that we're actually drawing node links using literally nodes and links, with points for nodes and connection marks to show the links between them. But what we're doing here is we're saying we're going to try to have an ordering within the view that mimics the subcellular localization within the cell, where we've got this very stylized way of seeing it. And then we're also going to try to cluster according to these functional groups. So there's different ways of arranging networks and trees, and actually drawing nodes and links is only one way of doing that. Now, what's interesting about this is we tried to have an algorithm that does automatically what we'd seen done by hand in a lot of textbooks and research papers, of having this stylized vertical thing where in the nucleus is at the bottom, outside the cells at the top, and we go along and show these subcellular localizations. But of course, we want to do this automatically rather than by hand. So it's this interesting question then of how can we mimic this with something that you can have it roughly the same size as what you would see in a textbook, and then also much, much larger sizes that no one would ever try to draw manually. Now, there's some other design decisions. Notice how we had these multiple views where we said, well, we'll call faceting the data across these multiple views. So we've got them side by side, and in all cases we've got the graph laid out, but we're color coding differently according to the experimental condition. So we've chosen this idea of chopping up the data across multiple views that are side by side. Why is that? So some people might think, well, what about animation? Can we just animate this? We have multiple time points, so it seems very natural if you've got multiple time points to animate them over time. But it turns out that the way the human visual system responds to things that change over time is really different than how it responds to things that you can see side by side. So here's our problem. Things are happening all over the place. This is only 8 frames. How bad could this be? And it's only about 100 nodes. But the kinds of animations that you think about as being incredibly evocative when you bring your kids to a Disney movie were carefully designed by people so that only one thing is happening at once on the screen and everything is drawing your eye to look at exactly the point where they're about to move. Lots of great techniques. There's a paper by John Lasseter about how to take the Disney animation techniques and automate them in computer graphics, if you're curious, from SIGGRAPH in the 80s. But we are not in this realm. We are in the data-driven realm where things can be happening all over the screen. So if we fixate on any one node, like let's take the one in the bottom left, orange, white, white, orange-ish, really deep orange. Okay, but what happened in the rest of the thing? We've completely lost track. So this is getting back to the idea of external representation versus internal memory. It turns out if you move your eyes between views, that's a perceptual act. In contrast, if you have to remember what you saw before and compare that to what you're seeing now, that requires internal mental resources, and it's actually harder. So that's how we can start to think about some of these trade-offs between side-by-side views and animations. And of course, it's interesting and complex, and at some point you can't keep adding side-by-side views because you run out of pixels. So there's trade-offs about the amount of pixels you have, the amount of internal memory and resources you're taking from the human, but that's the kind of thing where we can think about the design space now. So Cerebral ended up being the first system that really had this multiple juxtaposed views that really tried to solve the problem of interactively comparing the expression level new data to what was already known in the context of networks. And we proposed something that was new algorithmically, which is a framework for actually doing that kind of inspired by the hand-drawing layout in a way that actually scaled up beyond just the couple dozen nodes that people had been able to do with previous algorithms, because we had something that was a nice computational complexity class that was considerably more efficient than the previous work. If I had lots more time, we would get into that today, but we will not go down to the algorithm level today. Instead, I want to give you an overview of these different approaches to viz design problems. Just for the record, it was released as open source as a plugin for Cytoscape and is still available. Although there are some versions issues, hopefully we can get that working with the latest version of Cytoscape soon, although it's the version -1 at the moment. So that was Cerebral. Now let me talk about trying to solve quite a different problem, but using some of these same analysis methods and ways of thinking. So the MISB system was joint work with Mariah Meyer and Hans-Peter Pfister, and this was done in collaboration with some biologists at the Broad Institute in Boston. So once again, I'm going to start with a video to give you a sense of the look and feel.
MISB is a multi-scale Synteny browser for comparative genomics data. It is the first Synteny browser with side-by-side linked views of the data across a range of scales. The genome view showing all of its chromosomes, the chromosome view showing all of its blocks, and the block view showing all of the conserved genomic features within it.
Let me just make sure we've got that the right size. That's a little bit.
MISB incorporates a variety of visual cues and interaction mechanisms that allow biologists to understand 4 different relationship types: proximity, size, orientation, and similarity. The high-level genome view shows a circular overview of all source chromosomes on the outer ring, with the destination chromosomes on the inner ring. Selecting a source chromosome highlights it in black, and a copy of it also appears inside. We encode the syntactic relationship using connection, with B-spline curves linking the matched blocks colored by the destination chromosome using a repeating 8-element color map. The colors also provide an overview of all block destinations in the outer source genome ring. We can flip through the source chromosomes with left and right arrow keys or using mouse clicks. Our version of edge bundling is based on block contiguity and destination chromosome, making the location of spurious blocks more obvious. Changing the curve transparency makes the strongest connections stand out. We move the two triangles to filter the blocks in this view and in the chromosome view. Selecting a chromosome updates the chromosome view in the middle, which shows block locations in more detail. We can flip through the blocks with mouse clicks or using the up and down arrow keys. The selected block is outlined in the chromosome view and drawn in black in the genome view. The histogram track on the right shows the average similarity score for each block. The annotation tracks in the middle show where other features of interest occur, in this case, the tRNA and LTR transposons and conserved regions. Again, moving the triangles causes filtering in both views. Typing a genomic location into the Go-To box selects the nearest block. Selecting a block updates the block view on the right. The block view shows the conserved features, represented as oriented glyphs along the source and destination blocks. Matching features are connected with ribbons, which are pale red when connecting features with the same orientation, and pale blue when the orientations are inverted. We show statistical information about the feature pair similarity in two linked histograms, with bars next to the blocks for context, and also together for precise length comparisons. Mousing over a feature highlights the associated similarity value in the bottom histogram and also shows the feature ID. Analysis of blocks with many crisscrossings is made easier by flipping the entire paired block with the invert button. When there are too many conserved features to see clearly, the user can zoom in by double-clicking or moving the zoom slider and pan with the scroll bar on the right.
Okay, once again, I'm not going to show the entire video. That's enough to give you a sense of what the system feels like interactively. And so what I want to do is, again, talk about the whats and the whys and the hows. So what is the data here? Well, we've got this multiple scales of interesting information in these long lists or tables. So there's hundreds of thousands of features where we've got some idea that there's an ordered attribute. That's the position and chromosome coordinates. You guys are used to thinking about that. We've got categorical attributes, the orientation of the block, the chromosome on which it's got a matching feature, and then quantitative attributes of the length of the block and the similarity scores between the blocks. So while there's hundreds of thousands of features, there's actually, once you get down to the block level, there's thousands. And block is this somewhat fuzzy idea that's defined algorithmically by the researchers of some contiguous set of features on the same chromosome. And there's this idea of thresholds to say, is this a block or isn't it? And you want to combine them if the chromosome destination and source one actually have matched orientations and they're close enough together. And the question of what does close enough mean? That's exactly the question this visualization was designed to help people think about, is to improve and debug their algorithms for calculating these blocks, deciding whether they thought that what came out of their algorithmic pipeline was actually useful. So, and then again on the scope of things, dozens of chromosomes, and this was designed for comparisons between two genomes, either between species or within species. So let's talk about this in biology speak. In domain language, what would people say? They would say, well, we're analyzing the conservation relationship, meaning similarity, for those of us who are not biologists, between genomic features. And so what we noticed was, as we started talking to them about their tasks, there were really two levels of tasks. There's the reason they built the system in the first place, to answer some biological questions at the high level, like how long ago was there a common ancestor shared between each two species, or which segment of the genome might be responsible for function. But they also had all these much lower level questions that came down to, is the data correct? And they had to make sure that those were adequately answered before they could go start doing biology. So a lot of this came down to algorithmic questions like, are the paired features within a block actually contiguous, and which chromosomes share these conserved blocks, and are the similarity scores alike? So much lower level questions, and we wanted to build a tool that could address both of these. So putting on our visualization abstraction hats, we said, can we think about what is the common denominator of these biological questions that people are asking? And we ended up coming up with this idea of grouping the questions according to these ideas of, are they about proximity? Are they about size, or orientation, or similarity? So these were the categories of things the biologists were asking questions about. But if you'd asked them at the beginning, is this how you're thinking about it? This is not what they would have said. They had a very specific way of thinking about that we tried to abstract into ways coming at this sort of as third-party people from the outside. And there were these different scales of how much of the data do you need to see at once. The genome level, the chromosome level, the block level, the feature level. And there were these questions about, are you checking whether you believe your algorithm, or deciding whether these block thresholds are set correctly, or actually doing the high-level science? So there were these different levels of questions they were asking. So what we ended up doing in our final paper is really trying to go between their actual questions in the biological domain language and us having this more abstract way of thinking about this in terms of the relationships that mattered and the scale they needed to act at, as a way to justify our design decisions to see whether or not we were designing something appropriately. So let's talk about those design decisions. So you saw a particular way of showing the data where what we decided to do was encode these matches with both color in some cases and these literal connection marks in other cases. So what's the rationale for that? Again, I'm not going to go deep into the theory of visual channels in this talk. I'll just give you little glimmers along the way. But one of the ways to show that things are similar or different is to color code them by hue. And one way to show that things are actually linked together is to literally draw links between them to connect them. So what's the design space of ways we could do this? Those of you who've seen Circos know that there's this idea that you could have radial, you could have rectilinear, things could be either interleaved or next to each other. So this design space of how you could arrange, people had introduced various ideas about that in the previous work. The problem is if you have separate lines, once you start to get too many crossings, the visual clutter is so great you can't really see things. So there's some scalability limits on those. Well, what about if you actually tried to show these as separate things, and instead of drawing links, then you're color coding them? Sadly, this is an example of what not to do because of perceptual limitations in how we see color. Although those color-coded blocks on the right, you can actually tell the difference between pretty subtle shades of color. Once you have small, non-contiguous blocks of color, as you do on the left, mapping those from the mouse to the human chromosome, suddenly you're only able to tell apart about 7 or 8 colors. This is a fundamental human perceptual limitation, so sadly, you can't color code by chromosome when you're with humans. You have too many. So this is an example of some perceptual limits here. Once you start interleaving, things get quite complex. So we ended up proposing something where we actually had a mix at different levels, genome versus chromosome versus block level. We picked different answers about which things we're color coding and whether or not we have a linear view or a radial view. So we ended up with a hybrid solution. Now, getting back to this idea of side-by-side views, we have 3 views here. We have the large genome level view, then you pick a chromosome, and that actually filters out. Then we have the chromosome level view, and then we have a block level view. And these are all linked together. And one of the things that visualization research has shown is the power of having interactive highlighting between views, where if you highlight something in one view, it affects the others. And the thing to note here is we picked a different visual encoding idiom across these 3 views, so carefully chosen for the scale of the data that we wanted to look at. Let me mention again, there's some interesting trade-offs about how do you encode the data. When do you do things in a rectilinear way versus when should you use a radial encoding? Now it turns out, coming back to human perception, we are much more able to make precise comparisons when we have aligned spatial position than making perceptual judgments about angles. So there's actually some trade-offs about when do you use angles and when do you use rectilinear spatial position. Not going to get in deeply into that today. I just want to make you aware that there are some known facts about the accuracy of perception of these different ways to visually encode the information. There's also filtering. If you can't see everything at once, how do you usefully filter down the amount of data shown? We had some interactive filtering that you saw in the video. So what we ended up doing is on the outer ring, we summarized with color, and then once you select a chromosome, then on the inner ring, we show for that selected chromosome the relationships between that and all the others. We don't try to show you everything about the entire genome at once. The visual clutter would be too much. So we have this idea of selection of something of interest and then filtering. And in that inner ring, we're using both color and connection marks to try to make that relatively complex information usefully perceptually available to a human. So what we're trying to do is both identify and compare things and then summarize the whole. So MISBI was the first Symphony browser that had these side-by-side linked views that really supported an analysis at this different range of scales and tried to encode all 4 of these kinds of relationships. So again, open source and available on Mariah's site. So now let me get to the last bit. Tree juxtaposer, and in contrast to the first 2 systems, which were very much this problem-driven design studies, tree juxtaposer was much more of a technique-driven paper. It was this question of how do we actually scale up algorithmically some of this stuff. So this is joint work with Gambartir, Tacharan, Zhang, and Zhou, and appeared at SIGGRAPH a few years ago. And again, let me show you the video first. Tree Juxtaposer, a new tree visualization tool supporting scalable comparison and navigation of phylogenetic trees of over 300,000 nodes. Even small trees are hard to compare with visual attention alone. Linked highlighting of nodes and subtrees helps. Perfect matches for named leaves are easy to find, but computing the best corresponding nodes in the interior required a new algorithm that runs in n-log-n time at startup. Highlighting alone can still lead to false negative conclusions. We see 2 leaves highlighted in exactly the same spot on both sides, but their structural relationship to the rest of the tree differs. We can mark the exact place in the tree where structural differences occur using the results of the corresponding node computation. Subtrees underneath the black edges are guaranteed to be contiguous on both sides. The red edges show where a subtree from one side maps to a non-contiguous area in the other. Mouse over highlighting also allows us to check this property on the fly. Biologists call continuous subtrees a clade, and determining whether a clade in one tree is also a clade in the other is a recurring core question. When comparing these larger trees of 4,000 nodes, automatic detection and marking of structural difference is even more critical. Our new navigation technique, where growing one area leads to shrinking of all other places that don't share the rectangle's horizontal or vertical strip, is a new global focus plus context approach called accordion tree. We can manipulate areas that exactly encompass a subtree for structured distortion, or freely drag out a rectangle in space that defines an area that we resize. We turn on linked navigation between windows, which allows manipulations of one view to synchronously drive the corresponding changes in the other. Our best corresponding node computational infrastructure supports this functionality efficiently. Unmarked objects, drawn in grayscale, are dimmed according to their depth in tree so that the brightness level is tied to the distance to the root. This redundant visual coding helps users stay oriented despite the distortion-based navigation. Marked objects have a similar coding, where their saturation depends on current screen extent. Marking the differences does not help here, when most of the interior nodes are different because we compare a non-binary tree of 3,000 leaves with a binary one. Marking the metazoa subtree in blue and the Viridiplantae subtree in green shows that despite the difference in interior structure, the leaves are contiguous on both sides. Zooming into the Asca mycota group brings us to subtrees, or clades, that are contiguous in the taxonomy on the left, but are spread out forests in the binary phylogeny on the right. Our algorithm ensures guaranteed visibility of highlighted objects, even if they are currently subtending less than 1 pixel of screen space. Okay, enough of that one also. So again, let's talk about the data and the task, the why and the what and the how. So this data was trees. So trees are a special case of networks. Phylogenetic trees are interesting because when they're reconstructed, you've got the siblings not having an intrinsic ordering, and you've inferred the interior nodes with these unordered siblings. And so the specific task we were trying to support were biologists that were comparing the topological structure of these trees, often to try to compare the output of either different runs of the same algorithm with different parameters or comparison across reconstruction algorithms. So it turns out that a lot of the systems they had for looking at a single tree were not enough to try to compare two trees. Comparison is a fundamentally harder task, comparing two things, than browsing a single one. And you really need explicit support in your visualization when you want to compare rather than just look at one thing. And I mentioned this idea of deriving data along the way by transforming. One of the things we had to do was compute this idea of a best corresponding node between one tree and the other, which actually ended up requiring quite a bit of fun algorithmic work of how is it that we could do that. And that was crucial then for the interaction of the system to make it usable. Once again, you see side-by-side views. So these are these juxtaposed views side by side. We actually had linked navigation where moving something in one could cause moving in the other. And linked highlighting where if you highlight something in one view, you see its best corresponding one in the other. That's actually more complex than simply saying this is the exact match, but trying to find the best match across. So you might have noticed this sort of exotic way of navigating where when we dragged out part, what's called stretch and squish navigation, you drag out part and the rest actually squishes. Notice how nothing ever went off screen. You can't have this question of what's behind my head as I've moved my camera. You actually maintain at all times the context, but some parts are big and some parts are much smaller. If we wanted to get into the analysis of these particular ways of distorting the geometry, we could get into that. I'm not going to emphasize that too much today, other than to note that it's this complex combination of both filtering and aggregation that a lot of people have explored in Viz to try to look at these large complex data sets. So Tree Juxtaposer was the first interactive comparison system that actually showed the structural difference. For those of you interested in algorithms, I'll mention that there's a lot of nice algorithmic stuff to make sure that the drawing speed was dependent only on the number of pixels in the screen, not on the number of nodes in the tree. That's really crucial for scaling up to huge data sets. Again, also open source. At this point, the code is getting a bit old. But if people were curious to try to revive that, it would certainly be possible. So now that I've shown you these three systems, let me actually wrap up. So what I've shown you is I've analyzed these three different systems for biological visualization according to this way of thinking about what and why and how. And I've given what might be perhaps tantalizing glimpses into that without trying to slog you through the entire framework in the book, which is something I've written over a number of years to try to help people do this kind of analysis. And it does propose a whole structure of these questions of what are the data abstractions useful in visualization, why are people looking at that data in this very abstract way of thinking about the tasks in terms of trends and outliers and different kinds of searching and comparison and so on. And then how is sort of the heart of the book. What is the design space of visualization idioms, from visual encoding to using things like shape and color and size and orientation to these ways of manipulating data interactively and faceting it across multiple views and reducing the amount of data to show in a single view. So there's quite a lot there. I've hopefully at least maybe gotten you curious about things in that. So if people are curious, these talk slides are up. No need to be writing things down besides just that one URL or just going to my homepage and looking under talks, and then the one at the top is the VanBug one. There's a lot more on the pages for both my group and me personally of a lot of research papers, videos, open source software, a number of talks, entire courses. If people are curious about this book, feel free to grab. There's a 20% off promo code, which should work for the indefinite future. That gets more into these visualization analysis and design questions. And finally, let me acknowledge funding from both Agilent, NSERC, and NSF for these three projects. So thanks much, and I think we can take a few questions. Elodie. That is a great topic and one that is dear to my heart. It is not just the technical complexity. It's actually because of some fundamental human perceptual limitations. So if we had another 20 minutes or so, I could go on to a rather extended rant about what some of the issues are with 3D. But briefly, if you have data where you get to choose the spatial position, then usually it doesn't make sense to try to embed that in 3D because suddenly you're dealing with things like occlusion where some things are on top of the other. And yes, you could resolve the occlusion by moving it, but then there's that mental load of remembering what you saw before. An even deeper question is perspective distortion. So a lot of people think about the ability to actually draw things in perspective as one of the great crowning achievements of the Renaissance, right? We learn about this in our history classes and it's great. But when we're visualizing data, it's actually deeply terrible for the reason that the ability to make comparisons in the image plane in 2 dimensions, we're really, really good at that. Once we add depth, we actually are much less accurate at comparing things. So if you want to actually encode things with spatial position, which is the strongest perceptual cue, you torpedo a lot of that by going into 3D. Now, having said all that, that's if your data is non-spatial. If your data is intrinsically spatial and was acquired in 3D, then it turns out that the need to understand that 3-dimensional shape is completely central and crucial, and you really, really want to have 3D. So it all depends on the characteristics of your data. Is it intrinsically 3D spatial data, in which case you almost certainly need to have shape perception supported, and then interactive 3D navigation is really, really important? Or is it abstract non-spatial data where you picked how to lay it out? And in that case, it often gets pretty difficult to justify 3D. Not impossible, it sometimes does work, but you typically have to justify it carefully because often it causes more problems than it solves. Stephanie.
So for biologists in the audience, they want to talk to someone like yourself, and they're trying to phrase your recommendation. Someone's going to come and say, okay, I have this problem. Can I help you understand my problem? What do you recommend for developers?
That's a good question. Because my first thought is, well, it's our problem to try to interpret your language. You know, part of it is spend the time to actually talk to us so we can have this meeting of minds about when you're using these terms, what does that mean? So I think a lot of it is making sure that if you want to collaborate with a viz person, you've got the time to, it might take longer than you think is reasonable to come to some meeting of minds on basic terms, and you might think, you know, just go read a biology book, come on now. But there's this back and forth about trying to cast your specific problem into this abstract language and then checking back with you to make sure that we've actually got it right. So we typically do a lot of very iterative design and not just say, we talk to you once and then we go off for 6 months, design a tool, and then declare victory. There's usually much more of an engagement process where it's the time to go back and forth and talk to each other a lot that I think is a really crucial part of that. So I think it's if you're doing something that's not trivial, devoting the time to having that engagement back and forth in multiple rounds, I think is the crucial thing. Not so much any particular words you use in the conversation, but that process of continued engagement. Sydney. I think the answer is in between those two. So for those who couldn't hear so well, the question was about adoption and usage as a metric for success. So the short answer is there are many factors besides the goodness of the viz tool that affect adoption. So it's a really noisy signal. but it is a signal. And one of the signals I care about the most is, has anyone besides the designers of the tool used it effectively? One thing we want to avoid is something where the viz people used their tool, but they never actually deployed it. So for me, there's a big cliff between only the designers used it versus target users who are not the designers used it. And then I think another, and so you can actually have some very successful projects of the people that we built it for really did find something with it. And then there's another kind of jump, which is people beyond that set of folks that we originally talked to were able to effectively use it. So it's sort of migrating beyond that original kind of smaller set of target users. So that's another, and then I think, then when you start getting into, you know, some people use it versus lots of people use it, you get to a lot of these other factors. But this, non-designers and then people beyond the original target audience, I think those are some pretty interesting and useful signals. So it's certainly the case that it could be that it's terrible viz and it's the only place they can get that data, so they are going to use that tool by God because they have no choices, just like there's some really terrible tools that are in use because they're kind of, they've taken over the software ecosystem, Microsoft. So it's not a perfect signal, but it can tell you something pretty interesting. Yes.
So as a designer for the designer, that have you done any work trying to use your data visualization techniques to make very complex information accessible to the data?
Yeah, we have. Let me actually jump to this question of, are we enabling data discovery or are we presenting known information is one of the key questions about these goals. And so a lot of the same techniques can be suitable for both. But this question of exploration versus presentation is a pretty deep one. And so the good side is if you're presenting something known to a lay audience, you aren't so much trying to help them with this long process of exploration. On the other hand, if it's a lay audience, you typically don't assume that they have things like a bunch of domain knowledge that you can exploit. So you kind of win some and lose some in this context of presentation to the lay audience. Sometimes the discovery tools can be used then to present, but sometimes it's useful to actually try to think about tools just for presentation. So in particularly like health-related and biological analysis situations, I often distinguish between a researcher, a clinician, and a patient in terms of both their available time and their background knowledge. You know, the researcher can dig deep, the clinician often is extremely time limited, and then the patient doesn't have that domain background knowledge. So they tend to be three pretty different domain situations in my vocabulary, and thus you end up often with some different ideas about what's their task, which can then guide the visual representation. There's a question in the green vest.
So you mentioned that relationship could reduce the amount of time it takes per hour, minute. What if the tasks still take a really long time? I wonder if eventually using the is something neural net like that, where that watches you really.
So, yeah, so for those who couldn't hear, the question was, well, can we at some point, even after we've done everything we can and it's still taking too long, can we get the human out of the loop by using, say, deep learning techniques or something else? My answer would be a little different, which is typically that I think the really successful interactive visual data analysis tools are doing a mix of the human doing the looking and the system actually doing significant computation along the way. So what's happening is not simply that we're just laying out the data and then the user goes click, 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 and they're sort of mechanically going through and searching the whole possible set of things that the tool could draw for them. It's much nicer if we can have something where in response to some interactive choices by the user, then the system is actually going and doing a fair amount of computation in order to show them the next thing. So you could think about it, if you like machine learning analogies, as more of an active learning context where you get a little more information from them and then do a bunch of computation and then get a little, so we're not so much trying to take them out of the analysis loop completely, but we're trying to accelerate a lot of these kind of small micro loops in the analysis process. My take is that there's a lot of situations where if you just threw machine learning at a situation, you might get an answer, and it might tell you 0.63, and then you get to decide, do I believe this answer? And so it gets quite tricky to understand what is it that went into that data-driven decision? And for some situations, that might be good enough. You might be able to say, fine, I actually believe the machine learning solution, but I think there's a lot of, particularly with scientific data analysis, where you need to dig in and try to understand more of the details behind it, rather than just have a black box that gives you an answer. So if you trust the black box, great, but I think there's a lot of situations where you won't. You want to have maybe this interactive visual data analysis where you have a bunch of what I'll think about as, smaller white boxes scattered through, where you're doing some computation that's relatively understandable to the human, so they understand, when I selected this and now I'm seeing this. That's a small part of the analysis process I believe in, that I'm willing to trust, and then I'll continue on. So some stuff will get automated away, but I think a lot, particularly for exploratory data analysis, probably won't get automated away really soon. But you're talking to a viz person. You might get a different answer from an ML person. Yes?
Have you ever proposed the fact that you want to classify the data? Just because when going from out data in the graph, that's so sometimes I think about like data. And if so, how do you
So you use the word compromise data. You might use a slightly different word, which is to reduce the amount of data that you show. So I think as soon as you get to any kind of interesting real-world complex data sets, we almost certainly can't at every moment show all the data. And so we're often making decisions about what to show. And so I think particularly in that column where I'm talking about data reduction techniques and thinking about the trade-offs between, say, just filtering some things out or trying to somehow aggregate things together so that there's a proxy for a whole set of things that we can only show, a lot of that is a question about, if we can't at this moment show all the data, can we first make sure they understand what it is that we're not showing, and not being misled about what got filtered out or making the wrong inferences based on not understanding that we have filtered some things out. But at some level, almost all visual encoding is a choice of emphasis, a choice of, even if you're showing all the data, but you're showing it with different kinds of visual variables and some are really salient and others are not so much, that's already an editorial choice about what to show them. So in some sense, any visualization, whether you're showing all the data or only parts of the data, is your choice as a designer, or possibly as the user of the tool, making choices in the interface about what to emphasize and what to leave out. So in some sense, all visualization is this trade-off about what to leave out. And I think a lot of what we want to do is make sure they understand explicitly what was left out and not be misled, and try to help them get to the crucial part, because there's a lot of tasks, and actually going back to tasks, where sometimes you want to summarize all the data, but sometimes you want to pick a subset and just compare between that. And sometimes you really are just trying to find the one thing you're looking for. And so depending on their task, it might be very legitimate to filter out most of the data, because you're able to say for their task they don't need to see it all. Whereas for some tasks they really do want to see it all. Or maybe they want to interactively pick some interesting subset and then say summarize this subset. So I think there's a lot of nuances about how it is that you can choose what to show with a viz system. And the key thing is making sure it's actually meeting their analysis needs. Stephanie.
If someone wanted to get into this area, sort of from the outside, what kinds of undergraduate level courses would they take?
Yeah, it is an interesting field. So the question was, what kinds of undergrad level courses would someone take? And to a first approximation, my answer is it depends. There's a lot of different ways that people have come into the field. So like some folks come out of computer graphics. That was actually my background. Others come out of human-computer interaction. Others come out of, they're actually in a domain and they get sucked into building the tools more and more. So they were trained as domain scientists, whether it's biology or physics or whatever, and then they blinked and 10 years went by and they found themselves a viz person. Some people come out of math, particularly out of stats. Some come out of more, optimization, which is more maybe on the applied math side. Some are going to come out, I ended up getting sucked into this by talking to a bunch of hyperbolic geometers. This is not the usual path. It just happened to be the mathematical path that got me into there. So a lot of people have come from different domains and different backgrounds, and I think that's one reason we have this sort of diversity of paper types. So the fact that we'll take algorithmic work and very ethnographic work and controlled experiments on human subjects in the lab and deploying things in the field as in ethnographic things, and some very, very theoretical papers that come down to let's do a complexity analysis of this way of laying out nodes and links on a page, to, you know, let me really worry about the interaction design and make it so that it's easy to use. So, or let me worry about how it is that I could draw a million things at 30 frames a second when I only have this many pixels. So these questions about, do you have algorithmic concerns? Do you have more sort of problem-driven concerns? Are you doing a lot of controlled experiments or field study style experiments? It's kind of a pretty big tent where people can come in and do things with a lot of different backgrounds. So whenever people say, well, you know, even basic questions like what math should I take? It's like, well, which flavor of Viz do you want to do? Do you want to take a bunch of stats to be able to talk about controlled experiments in a lab? If you're doing some of the more geometric stuff, do you want to understand more about computational geometry? You know, do you need to know some basics of linear algebra for computer graphics? So there's a lot of different, you know, should you learn some graph theory if you're going to be doing a bunch of network layouts? So there's not a single answer. Having said all that, in a perfect world, I would love if students came to me having already had some human-computer interaction, some computer graphics, some statistics. You know, that's a perfect world. I often have a lot of students who come to my group where they have some kind of domain science background, you know, ranging from astronomy to biology. You know, people with graphic design and fine art training is also a pretty fantastic thing. It's rare that you get all of this in any single person. So I think that it is now 7:30 and everyone might be thinking about pizza. So perhaps we should wrap it up. So thanks very much.